السلام عليكم ورحمة الله وبركاته. تكلمنا في المحاضرة السابقة عن موضوع الأنتيجينز وعرفنا what is the antigens, what is the immunogen, haptens, and what is the antigenic determinant and definition of antibodies. تكلمنا عن factors influencing immunogenicity اللي كانت تتكون من contribution of immunogen, contribution of biological system, and mode of administration, and تعرفنا على chemical nature of the immunogen, types of antigen واللي احنا تكلمنا عن T dependent antigen, T independent antigen, and hapterin carrier antigen. وآخر شيء تكلمنا عن موضوع السوبر antigen. شفنا which bacteria or viruses that represent an antigen as a super antigen. وتكلمنا كذلك عن الانتيجين antibody reaction. Now we'll talk about the third structure, which include the complement system. Starts. What is the objective of complement? Understanding the teaching objective of complement system. First, to understand different pathways of complement activation, to know the enzymatic and non-enzymatic mechanism of complement activation, to know the biological properties of complement activation, to know the significance of complement system in host resistance, inflammation, and damage to itself, to understand the mechanisms of regulating complement activation and its product. So, what is the complement? Historically, the term complement was used to refer to heat-labile serum component that was able to lyse bacteria. However, the complement is now known to contribute to host defense in other ways as well. Complement can opsonize bacteria for enhanced phagocytosis. It can recruit and activate various cells, including polymorph nuclear cells and macrophage. It can participate in the regulation of antibody response, and it can aid in the clearance of immune complex and apoptotic cells. Complement can also have a detrimental effect for the host. It contributes to inflammation and tissue damage, and it can trigger anaphylaxis. Complement is actually composed of over 20 different serum proteins that are produced by a variety of cells, including hepatocyte, macrophage, and gut epithelial cells. Some complement protein bind to immunoglobulin or to membrane component of the cells. Others are proenzymes that, when activated, cleave one or more of the complement protein. Upon cleavage, some of the complement protein yield a fragment that activates cells, increase permeability, or opsonize bacteria. This table shows the complement proteins, which include the C1, C2, C3, C4 to C9, factors B, D, H, I, and proteins, mannose binding lectin, mannose binding associated serine proteins, the MSP1, MSP2, C1 inhibitor, C4 binding protein, decay accelerating factors, and complement receptor 1, protein S. All these complement proteins play together to exert their actions. هنا نتكلم عن موضوع ال complement system and their role in immunopathogenesis. راح نشوف في السلايدات التالية types of complement system and complement pathway activation, which may include the classical complement activation, the alternative pathway of activation, and lectin pathway of activation. راح نشوف الفرق بين activation of complement by the classical pathway, which is presented by antigen-antibody reaction, or the alternative pathway activation, which is presented by the presence of certain cellular antigenic determinant present within the bacterial cell wall, and the lectin pathway, where the antigen present on the sugar structure like mannose, which is a lectin that present on the cell wall. هنا different pathway فقط تتكلم على كيفية activation of certain type of complement proteins, with the result is the formation of the membrane attack complex. هذين الثلاث pathway يتحدن على شيء واحد وهو the C5 convertase. راح نشوفها بالسلايدات القادمة شلون يتكون C5 convertase by different type of complement activation، سواء كان antigen antibody reaction أو سواء كان عن طريق lectin binding or the alternative pathway. C5 convertase responsible for formation of the membrane attack complex. membrane attack complex من يتكون واللي final step in activation of the complement will result in cell lysis. A certain product of the activation product of the complement system mediate certain chemical reaction. It may include stimulation of certain cells like mast cells or basophils to release certain vasoactive substances like histamine, or it may mediate chemotaxis or anaphylactic reaction or vasodilatation, and these products will be represented later on. So there will be a classical pathway, lectin pathway, and the alternative pathway. The classical pathway is an antibody-independent pathway where there will be a binding of antibody to antigen and resulting activation of this pathway. The other type, as we talk, is the lectin pathway and alternative pathway. Both of them are antibody-independent. These different types of pathway result in activation of complement C3 and generation of C5 convertase. Activation of C5 will result in lytic attack pathway, where there will be formation of membrane attack complex, and the resultant is the lysis of the cells. So, in classical pathway, it will start with C1 complement, where consists of C1R, C1Q, and C1S. The trigger of activation of this product is the presence of antigen-antibody reaction. The antibody could be an IgM antibody or could be an IgG antibody. These complex will result in a cleavage of complement C2 into complement C2A and complement C2B, and cleavage of the complement C4 to C4A and C4B. The larger molecule of these C2-4B and C2A is the C5-C3 convertase. So there is a sequence of activation. لذلك احنا نسميه the complement activation pathway. In the absence of antibody, the binding of C1 to antibody is via C1q, and C1q must cross-link at least two antibody molecules before it is firmly fixed. The binding of C1q results in the activation of C1r, which in turn activates C1s. The result is the formation of activated C1QRS, which is an enzyme that cleaves C4 into two fragments, C4A and C4B. The C4B fragment bind to membrane, and the C4A fragment is released into the microenvironment. And we will see later on what is the role of C4A in the attractant and vasodilatation will result in inflammation. Activated C1QRS is also cleave C2 into C2A and C2B. The C2A bind to a membrane in association with C4B, and the C2B is released into the microenvironment, which exert a same action of C4A. The resulting is C4B C2A complex is a C3 convertase, which cleaves C3 into C3A and C3B. C3B bind to the membrane in association with C4B and C2A, and C3A is released into the microenvironment. The resultant is C4B, C2A, C3B is the C5 convertase. The generation of C5 convertase is the end of the classical pathway. So, several products of the classical pathway have a potent biological activities, and this resulted in the table 2. C2B is a prokinine cleaved by plasmine to yield kinin, which result in edema. C3A is an aflatoxin, can activate basophil and mast cell to degranulate, resulting in increased vascular permeability and contraction of smooth muscles, which may lead to anaphylaxis, and these presented by the release of histamine from these cells. C3B is an opsonin, promote phagocytosis by binding to complement receptor activation of phagocytic cells. It mediates phagocytosis. C4A is the anaphylotoxin. C4B is an opsonin. So, as we see, C4B and C3B are opsonins that will attach to the antigen and mediate phagocytosis. This procedure is under control condition. شنو معناها؟ احنا مو راح نخلي الكومبلمنت سيستم continuous activation. ذاك الوقت إذا كان continuous activation there will be a consumption of the complement system. لذلك there should be a regulator for these complement, whether it's activation or inhibition. So if the classical pathway were not regulated, there would be a continued production of C2B, C3A, C4A. Thus, there must be some way to regulate the activity of the classical pathway. And this is represented by table 3. The net result is the cessation of the complement activation, where there is no need to further attack of the invading antigen. The importance of this regulator protein in the classic pathway is demonstrated by the result of a deficiency in this inhibitor. C1-INH deficiency are associated with the development of hereditary angioedema. Those patients suffering from edema of the upper and lower lips or laryngeal edema, which is a serious condition. Another pathway is the lectin pathway. هنا فقط ترى التسميات تفرق. structure نفس السي 1 ترى لو تشوفون mannose binding lectin consists of mannose activating MSP1 and MSP2. These are the trigger for the activation of the complement system on the bacterial cells. Here the C2B to A is the C5 convertase. The lectin pathway is very similar to the classical pathway. It is initiated by binding of mannose binding lectin to bacterial surface. Here is the bacterial surface, and this is the mannose binding lectin. It is initiated by binding of mannose binding lectin to bacterial surface with mannose containing polysaccharide. بالclassical pathway كان activator is the antigen antibody complex. This type of activation in lectin is mediated by polysaccharide. The binding of MBL to the pathogen results in association of two serine protease MSP1, MSP2, and these are similar to C1R and C1S respectively. That's the structure. And MBL is similar to the C1Q. Formation of this complex, which is we call it the trimolecular complex, result in the activation of MSP and subsequent cleavage of C4 into C2A and C4B with the formation of C3 convertase. The C4B fragment binds to the membrane and the C4A fragment is released into the microenvironment. Activated MSP also cleave C2 into C2A and C2B. The C2A bind to the membrane in association with C4B and C2 is released into the microenvironment. The تشوفون mode of action in both classical and the lectin pathway is the same, لكن الاكتيفيتر فقط يفرق. These substance released into the microenvironment has the same action of substance released from the complement product of the classical pathway. Here is the generation of the C5 convertase, which is the activator of the membrane attack complex. The third one is the alternative pathway. Amplification loop of C3b formation. In serum, usually there is low level of spontaneous hydrolysis of C3 into product of C3I, factor B, bind to C3I and become susceptible to factor D, which cleave factor B into BB, and C3-1BB complex act as C3 convertase and cleave C3 into C3A and C3B. Once C3B is formed, factor B will bind to it and become susceptible to cleave by factor D. The resulting is C3BBB complex is C3 convertase that will continue to generate more C3b, thus amplifying C3b production. Now there is a usually spontaneous cleavage of C3 into their product under normal condition, and it is continuous process. The product of C3 من متلقى الانتيجين, it will be inactivated by binding to certain molecules. لكن إذا كان الانتيجين like bacterial cells is present, the product of C3b will bind to the bacterial cells, and there will be an amplification process where more activation of the C product. So the spontaneous production of C3b is tightly controlled with the control of amplification lobe. Spontaneously produced C3b bind to autologous host membrane and interact with decay accelerating factor, which block the association of factor B with the C3b, thereby preventing the formation of additional C3 convertase. Decay accelerating factor, it will prevent binding to factor B. This is in the normal condition. In addition, decay accelerating factor accelerates the dissociation of BB from C3b in C3 convertase that has already formed, thereby stopping the production of additional C3b. Some cells possess a complement receptor. Binding of C3b to complement receptor 1 facilitate enzymatic degradation of C3b by factor I. In addition, binding of C3 convertase to CR1 also associate BB from the complex. Thus, cell possessing a complement receptor 1 also plays a role in controlling the amplification lobe. Finally, factor H combined to C3b bind to the cells or in the fluid phase and facilitate the enzymatic degradation of the C3bb by factor I. Thus, the amplification lobe is controlled by either blocking of the formation of C3 or by dissociating C3 convertase by the enzymatic digestion of C3b. This is the decay accelerating factor, and this is factor BB, and this is factor H. All these processes is tightly regulated by CR1, factor H, and factor I. The importance of controlling this amplification loop is illustrated in patients with genetic deficiency in these factors, factor H or factor I. These patients have C3 deficiency and increased susceptibility to certain type of infection. Stabilization of C3 convertase by activator, protector surface. When bound to an appropriate activator of the alternative pathway, C3b will bind to factor B, which is enzymatically cleaved by factor D to produce C3 convertase. However, C3b is resistant to degradation by factor I, and C3 convertase is not rapidly degraded since it is stabilized by the activator surface. The complex is further stabilized by properdine binding to C3bbb. Activator of the alternative pathway are component on the surface of pathogen, include lipopolysaccharide of gram-negative bacteria and the cell wall of some bacteria and yeast. يعني من circulating micromolecules of the C3 within the serum without binding to the triggers, there will be no activation. لكن من ال C3 product bind to the component of the surface of pathogen like polysaccharide or cell wall of some bacteria and yeast, there will be activation and amplification of activation product. Thus, when C3b bind to the activator surface, the C3 convertase formed will be stable and continue to generate additional C3a and C3b by cleavage of the C3. Generation of C3 convertase. Some of the C3b generated by stabilized C3 convertase on the activator surface associate with the C3bbb complex to form C3bbb C3b complex, and this is the C5 convertase of the alternative pathway. كل complement system سواء كان classical أو lectin أو alternative pathway will end into C5 convertase. The generation of C5 convertase is the end product of the alternative pathway. The alternative pathway can be activated by many gram-negative bacteria, most significantly by Neisseria meningitidis and Neisseria gonorrhea, some gram-positive bacteria and certain viruses and parasites, and the result in lysis of these organisms by the formation of membrane attack complex will be talked later on. Thus, the alternative pathway of the complement activation provide another mean of protection against certain pathogen before an antibody response is mounted. A deficiency in C3 result in an increased susceptibility to these type of microorganisms. The alternative pathway may be the more primitive pathway, and the classical and lectin pathway probably developed from it. These are the structure of C5 convertase. Now, membrane attack complex, where all type of complement activation will end into the formation of the membrane attack complex when it comes on the surface of the antigen, can bacteria, gram-negative, gram-positive, or other pathogen. قبل ما نتكلم عن الممبرين اتاك كومبلكس راح نسوي سمري بسيطة حول الموضوع برمته. شنو الفائدة من وجود الكومبلمنت سيستم بالسيرم مالتنا؟ Certain product of the activated complement will mediate release of certain vasoactive substances mediated by plasma cells, by basophils and mast cells, or release of certain vasoactive substances like histamine. Some products are anaphylatoxin. Some products are chemotactic for certain phagocytic cells like neutrophil and macrophage, and these will accumulate at the site of the presence of the microorganism, whether by the classical pathway where there is the presence of antibody associated with antigen, or the presence of bacterial cells like lipopolysaccharide or cell membrane, or the presence of lectins like mannose binding protein within the bacterial cells. The net result is the formation of the membrane attack complex. راح نشوف شنو وظيفة الفورمation of membrane attack complex. The C5 convertase from the classical or lectin or alternative pathway cleave C5 into C5a and C5b. C5a remain in the fluid phase, and the C5b rapidly associated with C5, C6, C7 and insert into the membrane. C5, C6, C7. The C5b 6, 7 complex is referred to as membrane attack complex. Subsequently, C8 binds, followed by several molecules of C9. The C9 molecules form a pore in the membrane through which the cellular content leak and lysis occur. Lysis is not an enzymatic process. It is thought to be due to physical damage of the membrane. How? Non-formation of these pores, the water will enter into the cells. The entrance of the water into the cells result in cell swelling, and thereafter there will be mechanical damage to the swelled cells and disruption with the release of their content into the microenvironment. C5a generated in the lytic pathway has several potent biological activities. It is the most potent anaphylatoxin. In addition, it is a chemotactic factor for neutrophils and stimulate the respiratory burst in them. And it stimulates inflammatory cytokine production by macrophage. الصور C5 release at the site of inflammation. شو راح يصير؟ كل هالميكانيزم هاي راح يصير بها activation وكلها result in attack of the antigen. Its activities are also controlled by inactivation by carboxypeptidase B. إيه لأنه إذا كانت مستمر release راح يصير harmful effect to the cells. So at the end of their action there should be an inhibitor for this C5a. Some of the C5b 6, 7 complex formed can deassociate from the membrane and enter to a fluid phase. If this were to occur, it would then bind to other nearby cells and lead to their lysis. The damage to the bystander cells is prevented by protein S, vitronectin. Protein S binds to the soluble product of this molecule and prevent it is binding to other cells. So it prevents formation of membrane attack complex in neighboring cells. This table summarizes some of the important features of the complement system. Any classical pathway, lectin pathway, and alternative pathway. What is the component? Whether it is antibody initiated or not, whether it is initiated by pathogen in the surface, what is the cation required, procaine generation, anaphylatoxin generation, C3 convertase, C5 convertase, feed into membrane attack pathway, regulatory component. It will summarize all lecture in this table. Here, some biological activities of C2b, C3a, C3b, C4a, C4b, C5a, and the membrane attack complex, and what is the inhibitor or controlling factors, and what is the effect on the cells, and as I said, what is their biological activities. Here, this table shows complement deficiency and their resultant disease. Examples: absence of C1 INH result in hereditary angioedema by overproduction of C3b. C1, C2, C4 predisposition to systemic lupus erythematosus. Opsonization of the immune complex helps to keep them soluble. Deficiency result in increased precipitation in tissue and inflammation. In the classical pathway, the membrane mannose binding lectin disease susceptibility to bacterial infection in infant or immune suppressed. The mechanism inability to initiate the lectin pathway. The alternative pathway, factor B or factor B, susceptibility to pyogenic infection, lack of sufficient opsonization of the bacteria. Complement 3, susceptibility to bacterial infection, lack of opsonization, and inhibitory to utilize the membrane attack complex. C3, C5, 6, 7, 8, and 9. Deficiency result in susceptibility to gram-negative infection, inability to attack outer membrane of the gram-negative bacteria. Properdine deficiency, susceptibility to meningococcal infection and meningitis formation, lack of opsonization of the bacteria. These the controlling factors: H or I, C3 deficiency and susceptibility to bacterial infection, uncontrolled activation of C3 via alternative resulting in the depletion of C3. Thank you.